everyone. Delighted to introduce Seth Riley to you today. He's an adjunct in our department, has been a close associate with many of us in the department for quite some time, since 2000. Just a little history, he got his undergraduate degree in 1988 from Stanford, and then got the urban ecology spirit and moved to Washington, D.C., where he was a National Park Service scientist and the Center for Urban Ecology. And then after that, went to graduate school, UC Davis, like most of our faculty, graduate degree.
Only it was.
Cabal. Got a graduate degree working on bobcats and great foxes in Golden Gate. And then took a little step backwards, worked with Brad. Tiger salamanders and lower vertebrates. He hated that though. Working on larger mammals. He took a position in the National Park Service here in the Santa Monica Mountains in 2000. And since that time, he's really been an invaluable colleague to many of us, given opportunities to our graduate students to research in the Santa Monica Mountains and kind of spark this whole research boom on vertebrates and other species in the Santa Monica Mountains as an interesting test system in urban ecology. So, without further ado, Seth is going to be speaking to us today about long-term studies of wildlife and urban national park lessons and opportunities.
All right, thanks a lot, Bob. And that was a helpful introduction. So I'd like to start with this slide to just show, as Bob sort of intimated there, that I've been interested in urban ecology and urban wildlife my whole career, such as it is. So I studied raccoons in DC after my undergraduate work, and then, as Bob said, I studied bobcats. So there's a bobcat down there, and there's Golden Gate Bridge there at Golden Gate National Recreation Area as a graduate student, but then I've been since 2000 here at Santa Monica Mountains National Recreation Area studying wildlife in the park here. So our mandate with the National Park Service is to do our best to preserve the resources of the parks, and for me as a wildlife ecologist, that means the wildlife. So here's some quality wildlife habitat in the Santa Monica Mountains with downtown Los Angeles in the background. So you can imagine there are some different challenges in a national park like ours than you might have in Yosemite or Yellowstone or lots of other places. And so all of the work that we do on wildlife, and I'm going to talk about all of these different groups here actually, is focused on understanding what are the effects of urbanization and habitat fragmentation on wildlife communities. So here we are at the Santa Monica Mountains. There's UCLA right down here. You can see the Santa Monica Mountains, and then the Simi Hills, which are also part of the park, are completely surrounded by development in the Los Angeles Basin, in the San Fernando Valley, major freeways like 101 and 405, and then highly fragmented areas like here in the Simi Hills, where you have little bits of open space remaining, but they're surrounded by roads and development. Okay, so one thing that I just wanted to mention and I want to focus a little bit on today is, so I work for the National Park Service, and one potential limit with working for a particular place like a national park is all of the work that you do needs to be focused on that place, right? So as I said, our mandate is to preserve the resources in those parks, and so I can't go to the superintendent and say I want to go study wildlife in Borneo or Africa or Alaska. So that's a limit potentially, but I think it's also a pretty cool opportunity, because what it means is we can really learn a lot about what's going on in one place. So we've been able to study lots of different groups, and I'll talk about a number of these today, different carnivores and reptiles and amphibians. We've done a bunch of road studies and coyotes and lots of different things, and we can do that over a long period of time. Often, as everyone well knows, it's hard to do long-term studies. You get a grant for 3 years or 5 years, and it's hard to turn that into long-term studies, but in ecology and conservation, that can be really valuable, and that's what I'll hope to convince you of today. So we've been studying bobcats since 1996, so that's 19 years now. As far as I know, the longest continuous radio tracking study of one population of bobcats. Been studying mountain lions for 12 years now, and that's really the second urban mountain lion study. Paul Beyer had studied them in the 90s in the Santa Anas, but there's others now, but it was one of the first ones. And then our amphibian and our terrestrial herp projects are part of our long-term monitoring program, so I'll talk about that a little bit. We've been doing both of those for 15 years now. And I won't have a chance to talk about it today, but we did a 9-year study of coyotes, one of your longest urban coyote studies, and then we've done a whole bunch of work on roads, particularly together with Caltrans. And I won't have time to talk about that today, but that's really a valuable long-term thing as well. Okay, so in terms of carnivores, as I said, we've been studying them since 1996, especially bobcats. And our goal is to understand how are these animals making it in this urban fragmented landscape. And so we've been studying lots of different aspects of their ecology and behavior, and I'm going to talk about a number of those today. So first of all, with bobcats, as I said, we've been studying them the longest. We now have close to 320 bobcats that we've handled and over 180 that we've radio tracked. So it's a lot of animals over a long time. All right, the first few years of the study, we learned some interesting stuff. One is that the home ranges were very small and the densities were high. So this is the first 4 or 5 years of the study, and you can see these home ranges for males and cats. Home ranges are typically bigger in males than females, about 3 square kilometers and about 1.5 square kilometers in females. Those are tiny. Those are basically as small as you'll see in any bobcat study anywhere that anyone's ever done. The southeast, and there's one study in San Diego that were similar, but so very small home ranges leading to very high densities. One of the things that we saw, again, in the early years of that work, and this is partly through some genetic work that we were doing here in Bob's lab, is that we saw a lot of overlap between females. So typically in cats, and in particular in bobcats, you have a lot of territoriality in the females. So for example, this is a female here, and you can see she's not overlapping at all with this purple one here and only a little bit with this green one here. And that was typically the case in other studies where females are very territorial. Males tend to be a little more overlapping of different males and different females, but we found a very different thing. So you can see these two females right here completely overlapping each other, not only their home range, but if you look at their core areas, which is a measure often of the areas of most intensive use, that they're totally overlapping each other. And through the genetic, we were able to see that those are closely related individuals. So their relatedness values were between 0.25 and 0.5, so they tended to be close relatives, half-siblings, or mother and daughter. So that was some interesting things we saw from the beginning, is high densities and pretty intense overlap even between females, which typically don't do that. So one of the most important things we saw in the bobcat study, so we've been able to, like I said, do it over many years and see changes in the population. And starting, well, for the first 6 or 7 years, the main source of mortality for bobcats was vehicles. But starting in late 2001 and mostly in 2002, early 2002, we started to see cases of this disease, which is mange. It's caused by this mite here, Notoedres cattai. And the bobcats get very sick eventually and emaciated and eventually die. And it was interesting, so I had studied bobcats in the Bay Area for many years and not seen this, and we've been doing it for 6 or 7 years here in the Santa Monica Mountains and never seen it. So the first few cases of this, we took them to the state lab and had them do a full necropsy, the California Animal Health and Food Safety Lab. And all of these animals that had mange were also exposed to anticoagulant rodenticides, so rat poisons that people are putting out to kill rodents. And the vet made an argument to us that maybe they are being compromised by being exposed to these toxicants, and that's making them more potentially susceptible to mange. So we saved up, this is for those first few years, we saved up all the livers we could find, and we tested them for animals that died from whatever, roadkill or mange or various different things. And we found, at least statistically, that was true. So here you see of animals that died of mange, 17 out of 19 were exposed to at least 0.05 parts per million of these rodenticides, whereas of ones that didn't die of mange, only 8 out of 20. This is for the first few years, and then through 2008 we were seeing that same pattern. So 22 out of 26 that died of mange had at least 0.05 parts per million, whereas only 9 of 21 that didn't. And so it seems like potentially, at least statistically, there's this strong association, and we've continued to see this association in more recent years. Now, what about that mange epizootic? It had a huge impact on the bobcat population that we've been studying. So these are survival rates for bobcats. So this means that the chance of a bobcat surviving in 1997 from one year to the next was 83%. And these are very high survival rates. So the survival rates were about 75% or more for those first few years, partly because there's no hunting and trapping, which is a big source of mortality in lots of populations. But you can see the number of deaths increased, and in particular the number of mange deaths. We had 1 in 2001, 9 in 2002, 11 in 2003, and the survival rate dropped from 75% to 50% to under 30% in just a couple of years. We also saw this. We were collecting scat for diet for a number of years, and then we kept doing it because it was valuable for getting an idea of relative abundance and distribution. You can see the number of scats we were collecting on these transects in late 2002, early 2003 just fell off the table here, and we have very few scats in many of those places all the way up through 2008. And then through the radio tracking data, we could see that as well. So this is a really interesting area. This is Rancho Simi Recreation and Park District area in Agoura Hills. So this is Canaan Road here. And we had 6 different bobcats we were radio tracking in here in 2003, 2002, and 2003. 5 of those 6 died of mange, and the 6th one we lost track of. And basically for a long time we saw no evidence of bobcats from scat or from trapping or from our radio tracking using that patch. So the big impacts of this mange on the population. One thing that I want to mention that we've been seeing relatively recently, and again, this is one of the advantages of doing this stuff over the long term, is we've started to see bobcats return to that patch of habitat. So these are locations for bobcats 2001 before mange. You can see we had these high densities, and then we had those 6 different bobcats. This is in 2006 after mange, so all those animals died. And you can see we had almost no use of this patch. A couple males would cruise through there once in a while, maybe looking for females. And it's not like we weren't trapping in there. We were trying to catch animals and we just weren't finding them. But in the last few years, especially starting in 2009 and 2010, bobcats started to make it back into this patch and even reproduce. And now we have a number of females and males that are reproducing in this patch. So that's been pretty interesting to see the return in that particular spot. And as I mentioned in a second, we know something about the genetics of that as well. Okay, so as Bob mentioned, we've worked with lots of different folks, graduate students and postdocs and things over the years. And that's, again, one of the things I think that's really valuable about long-term studies like this is it can really be helpful to have postdocs and graduate students come in and expand on and add to what we do. So Laurel Sirius, who was a graduate student who just graduated this summer of Bob and of mine, made amazing contributions to the Bobcat project in a number of ways, and a couple of them I'm going to talk about. One of the things she did, she actually expanded where we were tracking, where we were sampling animals way out east of 405 and out to Griffith Park, and actually found a bunch of animals with mange here in the Beverly Hills area. But the other thing that she did, which I'm really excited about, and this is just some pretty recent stuff, is she looked in much more detail at the health, physiology, and even immunology of bobcats, which was a huge amount of work. No one ever wanted to do immunology on bobcats before. But you found some really interesting things, and I'm not going to go into too much detail here, but just can talk about it more later if someone is interested. But basically what she found is that bobcats that were exposed to these rodenticides had this generalized inflammatory response, and we think, our hypothesis is that that makes it very difficult for them to respond specifically to the mange. So these are cytokines. She measured cytokine expression, and the purple bars are bobcats where they weren't exposed to rodenticides, and the orange ones are ones where they were, and there were lots of these different cytokines. So cytokines are small proteins that are cell signalers, right, that are very important in the immune system. And a lot of these are significantly higher in animals exposed to rodenticides than those that aren't. And all of these ones with arrows are actually cytokines that are involved in a general inflammatory response. And so Laurel uses this analogy, which I kind of like. So it's the plane here is like your immune response, and this guy here is like the cytokine, and he's going to guide the immune response to the target infection. But what we think is happening here is this broad scale general inflammatory response. And so the immune system is not able to, because it's broadly cranked up, it's not able to target the particular mange. And like I said, there's some more details about this if you're interested. But anyway, this is really interesting and really valuable too, because not everyone has been convinced of this association that we've been seeing, at least statistically, between mange and rodenticides. And so this has been super valuable to really dig into this this mechanism more. Okay, another thing that we've been interested in for a long time, of course, roads in Southern California are huge, especially freeways. And so a lot of work we've done together here with UCLA as well, it's been focused on what are the impacts of roads, both in terms of movement and behavior as well as genetics. So from the very beginning of the study, so this is still focusing on bobcats here, we saw that clearly the freeway was a major impediment to movement. So these are many different home ranges for bobcats over many years, and you can see they're all either on one side of the freeway or on the other. So clearly we were seeing movement-wise that it was a barrier, but it wasn't a complete barrier. So we've done some work. This is work of a master's student at Northridge, actually, where she was monitoring various culverts, and we did find some movement, including by bobcats, under the freeway. So then we were interested to know, okay, there's some movement, but not a lot. Is the restriction enough to cause changes in gene flow? And so again, that's work that we did here in the Wayne lab with using microsatellites, which are nice because they evolve quickly, and so we might be able to see something happen over a short 50 or 60 year period since the freeway was built. And we saw, in fact, which is somewhat surprising, I thought, and we saw this, I'm just going to mention bobcats, but we saw it for coyotes too, but we compared populations on either side of the freeway, so the actual physical distance here is very short, to populations on the same side of the freeway, and we found significantly more, these are FST values, significantly higher differentiation across the freeway than along the freeway, so about 0.05 across the freeway here. This one is non-zero, actually, which is interesting to note, so there may be some effect of the Canaan Road and the development here, but significantly more differentiation across the freeway than along it. And we saw this population structure evident in results from the program structure as well. I'm just gonna use these kind of figures a bunch here for a bunch of different species, and I assume people are largely familiar with it, but just to mention, so structure takes your genetic data and decides how many genetic clusters it thinks you have there, and then you can impose your geography on that and see if there's an association there. So this is for the bobcats. This was bobcats in Golden Gate, so fortunately they're their own cluster, which is good news. But the other thing that was interesting, just like we saw with the FSTs, this is the animals south of the freeway, and you can see they're almost entirely in this red cluster here. And then the animals north of the freeway, so this is to the west and this is to the east, were a kind of a mix of these green and blue clusters. The other thing that was kind of cool is where we can, again, put together different kinds of data over a long term, is we were able to look at some of these animals where they were caught north of the freeway, but they were genetically assigned south of the freeway. And there are potential migrants, and of these 13 animals, like these ones, and these ones, we had telemetry data on 12 of those, and 9 of those 12 either crossed the freeway or lived right near the freeway. So there was sort of extra evidence that maybe they were potential migrants. Okay, the other genetic data that we have comes from Laurel's work. So she, as I said, she majorly extended our sampling out to the entire Santa Monica Mountains and out here east of 405 and out to Griffith Park, and found some really interesting things. So this is another structure figure based on Laurel's data going all the way out to 2012. So one thing we found is here's 101, so these are north of 101 and south of 101. So again, we see the effect of 101, which is good. She also found significant effects in structure of 405, another major freeway. And then the other really interesting thing is these are bobcats from before mange, and these are bobcats from after mange. And you can see they're in totally different genetic clusters, which I think was really interesting. So the FST values, again, she saw 0.04, which was significant across the freeway, very similar, which is, again, sort of nice to what we had seen earlier. An even higher FST value across 405, so also a major barrier. And then actually the difference between bobcats in the same area before and after mange was 0.06, so even more than you're seeing across this freeway here between these two populations before and after the disease, which I thought was really interesting. And basically what's happening, so this is structured just with those animals north of the freeway, and you can see early on they're all in this green cluster, and by the end they're basically all in this red cluster. So we have, at least genetically, we have a new bobcat as a result of this mange epizootic, and she found significant evidence of a bottleneck. The one other thing that she did, and I'll just very briefly mention this, but it's really cool, I think, is she used non-neutral loci as well. So she used some MHC loci, and we saw very different patterns. So again, here's the structure diagram with the neutral loci, and here it is with these MHC loci that are associated with immune function. And you can see basically no pattern of differentiation, even with these major barriers and freeways and before or after mange. And so it looks like there's evidence of balancing selection from in these MHC loci. Just the last very quick thing I want to mention about bobcats, which again I think is kind of cool to be looking at over the long term. Every spring, mid-February, we start to radio track the females intensively, and we look for them to be over and over again in the same spot. And then we go in there and we try to find that spot and we look for kittens and evidence of reproduction. So that's critical, obviously, right, for populations and conservation. One of the things we're interested in, again, is what are the effects of urbanization. So these are females from fragmented areas and from unfragmented areas over a number of years. And actually, you can see there really wasn't any difference here. So about 2/3 of the females were reproducing even in the fragmented areas. So that was kind of good news from a conservation point of view and interesting to see. One of the things we've been seeing just in the last few years, though, all right, so here's 97. There were a couple years where we weren't doing the female work, but 97 through 2011, again, about 2/3 of the females are reproducing every year. You can see there was a number of years during the mange when we didn't have as many. But just in the last 3 years, we've actually had quite a few females in these areas, 7, 9, 10, and almost none of them have been reproducing. 2 different years with 0% and 1 year with just a couple that reproduced. So it's interesting to think about. I mean, these are years of major drought, right? And so there may be an effect on the rabbit population, which is the major thing that the bobcats are eating. So that's been pretty interesting to think about what might be going on there. And again, something that's valuable from these long-term studies. Okay, the other carnivore study that I want to talk about today is mountain lions. So we've been doing them for about 12 years with GPS collars and radio tracking kittens. We have data on 35 lions, so a lot fewer, an order of magnitude less than the bobcats, but that's partly because there just aren't that many mountain lions. So mountain lions are really sort of the ultimate challenge for conservation, we think, in a landscape like this. You know, they're really deer specialists, and though bobcats move over big areas, mountain lions move over. So a female mountain lion's using like 100 to 150 square kilometers as opposed to the 1.5 to 2 that the female bobcats are using. And so again, we're trying to understand how are they doing, what are they doing in this kind of landscape, how are they using it, but in particular for the mountain lions, it's especially important to understand are remaining natural areas effectively connected, what are corridors, and then this is one I'm not going to talk about, but that of course is important. Can people and lions coexist? And ultimately, can we do our job, which is to preserve lions in this park in the long run? And from the beginning, our study area and our whole sort of landscape of questions was very different for mountain lions. So this is the area that we were intensively studying, the bobcats and coyotes here. This entire thing is our study area for mountain lions. We assumed from the beginning that the Santa Monica Mountains by themselves would probably not be big enough for a viable population of mountain lions. And so these connections across 101, across 118, and ultimately across 126 to Los Padres National Forest would be critical. And we saw that from the very first few animals that we followed. So this is just from the first few years of the study. This is P1, the first male that we followed, and you can see he's using the entire Santa Monica Mountains from Point Mugu State Park in the west to Topanga State Park in the east. And that it's not an unusually large home range for a mountain lion. And then here's the first female we followed, P2. And sometimes females overlap in mountain lions, just like we saw in the bobcats. That's actually better known in mountain lions. And you can imagine there's room for maybe another female here and another female here. And if you want to be really generous, say there's 3 females in each of those areas. And maybe sometimes the males are splitting the mountains like we think they're doing now. There might be, maybe there's 2 or 3. You're still talking about less than 15 mountain lions, which is clearly not enough for a population genetically or demographically. So that means these connections across the roads are going to be critical, these freeways. And so what about mountain lions and roads? They're definitely getting back and forth across all the major north-south secondary roads to the mountains, like underneath here at Malibu Canyon Road, or over here at Mulholland. This is young female P23. But again, the freeways are the critical ones. So what about the freeways? And the freeways are major barriers. There's one freeway, 118 right here, that we have seen a bunch of crossing. 2 animals crossed multiple times over a couple years early in the study. But there's also a nice big hiker equestrian tunnel here. There's P3 with his collar there going through that tunnel, and they use that most of the time, if not all the time. But the critical one, 101, is a huge barrier. So these are some of we're up to 80,000 or so GPS locations over the course of the study, and you can see that the animals are almost entirely on one side of the freeway, generally south, or the other. So here's a bunch of points at the west end of the mountains, right up against the freeway. Here's a bunch of points in the middle of the mountains, again, up to the freeway, but not across. And then here in the east end of the Santa Monicas, you can see, and this is a problem in lots of places in Southern California, you don't even have natural habitat even up to the freeway for the animals to even get close. So they're up into the natural areas, but not even up against the freeway. Same thing with 5. We don't have as many animals that we've tracked along 5, but none of them have crossed. Here you have a number of different lions you can see right up against 5 and never going across it. And then same thing with 405. So here we are at UCLA right there. We have 6 different lions here. with lots of points right up against the freeway, but never across it. So the freeway is a major barrier, again, for movement for the mountain lions, and so we had the same kind of questions about what about population genetics. And so again, this is work we did with folks here in Bob's lab, so Bob and John Pollinger and Laurel, and then also with Holly Ernest's lab at Davis. Now she's at University of Wyoming because she had genetic data on mountain lions from all over the state of California. And also with mountain lions, and in fact even more strongly than with bobcats, we are seeing significant genetic effects of the freeway. So this is just the basic genetic data. Obviously you don't need to worry about all these numbers, but these are just different animals. Unfortunately, sorry, this is cut off a little bit over here, but these are animals. This is like 101 here, and these are all animals north of the freeway. These are animals south of the freeway. These are different loci. And what I've highlighted in yellow here these are different alleles that we found in animals north of the freeway and never south of the freeway. And overall, so we typed 42 animals at 54 loci, and of 172 alleles, 67 were never found south of the freeway, and 78% of those loci had these private alleles north of the freeway. And we saw some strong structure with program structure. Also with the mountain lions, even stronger. So these two especially, but all of this is some pretty strong structure. So these are all the mountain lions south of 101, and you can see they're very heavily assigned to that green cluster. And then east of 5 as well, they're very strongly assigned to this red cluster. And then this blue one is animals north of the freeway and then west of 5. And that structure has had an effect on the genetic diversity. So we compared animals north of the freeway to south of the freeway, and in all these different measures, so expected heterozygosity, numbers of alleles per locus, expected number of alleles per locus, the percent of loci that are polymorphic, there was significantly higher genetic diversity north of the freeway than south. And in fact, in the Santa Monica south of 101, the lower diversity than all other contiguous populations around the state, and basically in all other parts of the West. There was another population that folks are studying down in the Santa Ana Mountains that also had similarly low diversity. And so that's sort of interesting because it's also a totally isolated population. So extremely low diversity, lower than people have found anywhere else except for in Florida, where you have endangered Florida panthers that had very serious inbreeding issues. The other thing about, again, doing this long-term work and then putting it together with the genetic data that we got together with folks here at UCLA and at UC Davis is we were able to do things like put together this pedigree, and we saw one of the things that's contributing likely to the low genetic diversity is very close inbreeding. So right here is one event, and here is another event where fathers have mated with their daughters. So for example, P1 and P2 mated and had 5, 6, 7, and 8, and then later P1 and his daughter P6 mated. We were able to tell that through genetics, and then the same thing we saw P12 mated with P13, had this litter of 17, 18, and 19, and then P12 ended up mating, it looks like multiple times actually, with P19, his daughter. So that's clearly not beneficial from a genetic diversity point of view. Okay, so that's the bad news. Then we did have an exciting event though that happened. So for the first 9 or so mountain lions, none of them had crossed 101 ever in either direction. But in late 2008, we caught a young male in the Simi Hills, P12, and he crossed the freeway right in the Agoura Hills area at Liberty Canyon, which is an area we're very focused on. I could talk more about that if people are interested. But he crossed the freeway in early 2009. And fortunately, not only did he cross the freeway, but he survived. Right? If he had just made it across and then died, that wouldn't have helped us at all genetically. But he survived, and he successfully became a breeding male, which generally there's very few males that actually successfully become breeders in these mountain lion populations. And so here I have that same slide, but here was P12 before here north, and now he's south here. And you can see he brought some of these alleles from the north to the south, and then he transferred them to his offspring here, P17, 18, and 19. So he had 20 private alleles at 10 different loci, or northern alleles, and he passed them on to 19 of those alleles on to his offspring. And so now here's structure with P12's offspring. It's pretty interesting here. So here's P13, the female that he mated with, and you can see she's very strongly in this green cluster. And then here's P12 strongly in this blue cluster. And all the offspring are basically exactly 50-50, those two clusters, which is really what you'd expect, except for P23 and P24. And those two are the result of the close inbreeding. So P12 is not only their father, but also their grandfather. So you would expect about 75% assignment to this blue cluster, and that's exactly what you see. So we see these important events, so these inbreeding events, and then the important outbreeding when P12 crossed the freeway and mated with P13. And we were able to see that these individual events were definitely having these, in this very small population, we're having these population genetic effects. So this is some work that John Pollinger did, gene drop, with the program PMX, and he saw with, for example, gene diversity declined when the inbreeding happened, increased with P12 coming across, and declined with another inbreeding event, and then both mean kinship and the inbreeding coefficient did the opposite, so they went up with the inbreeding, down with the outbreeding, and up with the inbreeding. So these individual events are having these population genetic effects. So the last thing I want to talk about with the mountain lions is dispersal, which again is critical. So we're really concerned about our animals getting back and forth across the freeways and the roads, both in and out. And so over the years, again, we don't have huge sample sizes because these are just very low density animals, but we have 15 young males that we know of in the Santa Monica Mountains since 2002. And not one of these 15 have successfully dispersed to a new home range or to a new viable home range. There's one exception that did disperse, which I'll mention. And normally every single young male in mountain lion populations disperses and goes somewhere else, and half the females actually. And of the 12, there are a few that we lost track of, but of the 12 with known fates, 11 died before or during dispersal, some of them on roads like this on Malibu Canyon Road here. This young male was killed by police in Santa Monica. But the biggest source of mortality was intraspecific conflict, so specifically males being killed by other males, specifically adult males. And overall in our study, we have 14 deaths of collared animals, 12 of which we know the cause, and half of those were this intraspecific strife, so mountain lions killing each other. This is a source of mortality that you see in other populations, but we think it might be particularly exacerbated here because the young animals, as I said, are never able to disperse out of the Santa Monica Mountains. The other thing that was really interesting that we were able to see again from looking together with the genetics that Bob was pointing out actually is that a number of basically these animals are, so males kill each other in, and sometimes even females in other populations, but what's happening in this population is males are killing their sons and even their daughters and sometimes their brothers. So they're killing close relatives, and even in one case, male P1 killed a female that he had mated with, right? So all of this seems like a bad strategy, right, evolutionarily. And what we think is happening is that normally, like I said, most of the young mountain lions, all the males and half the females are dispersing, and they're just not able to do that here. And we've seen some evidence of that again through the radio tracking data. So here's a young male P8 who's going right out to the edge along 405 and up in the valley here, and then another male that went to the east, and you can see he was right up against the freeway and even out into the agricultural areas here in Camarillo. So, you know, we can't read their minds, but it looks like these males are trying to disperse. Here's a young male P14 right out to the very edges of the park here, and this crazy point over here down south of Sunset. So they're going right out to the edges, but not successfully dispersing somewhere new. And so we think they're maybe trying to avoid these adult males, but just not able to do that because of the development on the roads. So the last thing, just briefly about mountain lions, the one male that did successfully, we think, disperse out of the Santa Monica Mountains is P22. So here he is with the Hollywood sign in the background. He lives at Griffith Park, which is pretty amazing for a mountain lion. Some collaborators with USGS, part of the Griffith Park connectivity study and Cooper Ecological, were monitoring various crossing points near Griffith Park to see if animals were getting in and out, and they found a picture of a lion in February 2012, and then we were able to catch, this is Jeff Sikich, who's our primary field biologist on the lion project. He was able to capture him over here in Griffith Park about a month or so later, and we've been radio tracking him ever since. You can see he is all over Griffith Park, as much of it as there is, especially north of Mount Lee here, where it's relatively low density human use, although it's just people everywhere in Griffith Park, as you know, if you've been there. But the interesting thing to see is that this is P22 here, and you can see he's very clearly in this Santa Monica Mountains genetic cluster. And so if he came from the Santa Monica Mountains, which genetically it looks like he did, that means he had to cross two freeways, 405 and the Hollywood Freeway here, 101, to get to Griffith Park. Although now he seems to be relatively stuck there, so he's been there for 2.5 years now. in a home range of 26 square kilometers. So normally males are using 300 or 400, even more than that, square kilometers, and he's using 26. And as far as we could find anywhere in the literature, this is the smallest by quite a bit annual home range for an adult male mountain lion. What in theory should happen is what we saw up in the Santa Susana Mountains. So here's adult male P21, and actually we saw again with the genetics that P16 was his son, and you can see they were overlapping quite a bit. And then at some point P16 dispersed out along the Santa Clara River, and 126 he crossed over here between Ventura and Santa Paula, dispersed way up here by Mount Pinos, but then he found a home range which he used for multiple years before we took his collar off, actually, right here north of 126. So that's sort of what you would expect to happen if you had enough connectivity between these areas. Just mention this very quickly. This is sort of an idea that we suggested in our recent paper, but it seems like there may be sort of an interesting thing going on here. So an Ali effect, you remember from population biology, is where in very small populations you have low growth rates and various problems because of very low density. So animals, they can't find mates, they can't form social groups, there's various kinds of social disruption. But what seems like maybe is going on here is it's a small population, but it's highly constrained. So it's sort of a inverse or reverse Ali effect where the population issues are caused by high density or being circumscribed by these freeways as opposed to by low density. And so we're seeing social disruption because of interactions in this case that would otherwise probably be rare or non-existent, like males killing their sons and their brothers and their daughters, and males mating with their daughters. Okay, so the last thing I want to talk about is a couple of our other programs, so not carnivores anymore, but reptiles and amphibians, and this is part of our inventory and monitoring program that's occurring in the Park Service generally. So there's been a big move over the last decade or so to find out what's in the parks and then monitor it. And in particular, we pick what we call vital signs of ecosystem health. And for us in the Santa Monica Mountains, in terms of animals, we picked reptiles and amphibians because it's a big, diverse group, and there's a lot of interesting potential things going on and potential issues in terms of conservation. So we've been doing long-term, first inventory and then long-term monitoring of both terrestrial and aquatic reptiles and amphibians. So first of all, in terms of terrestrial, so we use these pitfall trap arrays with pitfalls and drift fence between the traps, and then these funnel traps to catch snakes. And we're trying to understand throughout the whole park, what is the distribution and abundance of reptiles and amphibians? And then again, we're always interested in what's going on, what kinds of things are affecting that distribution and diversity, and in particular in this kind of a park, especially urbanization and fragmentation. This is some of that intense fragmentation where we've had some of our pitfalls. So for the first 10 years or so of the study, we had pitfalls in 3 different areas. So Point Magoo State Park out here in the west end of the mountains, the least developed part of the park. In the middle of the mountains here, mostly in Malibu Creek State Park. And then in the Simi Hills here, and these arrays we were trying to use specifically again to look at the effects of fragmentation. We have 34 native species on our reptile and amphibian list, and 25 of which are largely or entirely terrestrial and therefore likely to be things we could detect reliably in our pitfall traps. And over just the first 8 years of the study, we're actually able to detect 24 of those 25 species. So that's good news, A, for the park. It means that basically we still have the things that we think we should have, and that we're able to detect them with this method. The only species we haven't detected actually is the lyre snake, which tends to like sort of rocky areas where it's much harder to dig a hole and put your pitfall. But generally we have the species that should be here. This is just an excuse to show gratuitous, cool herp pictures, but Ensatina and mountain king snake, horn lizard, night snakes, and patch-nosed snakes. So all of these things are things that we are finding in the Santa Monica Mountains, and that's good news. But what about the potential effects of urbanization and fragmentation? So again, in the Simi Hills, we put pitfalls in some core areas like here and here, and then in large habitat fragments, and then in very small habitat fragments to see what kind of things are we seeing in terms of abundance, diversity, and distribution across the landscape. And we saw right away that certain species are being lost in those very small patches, especially larger snakes. So you can see rattlesnakes we were never getting in these small patches. And then same thing with striped racers here. So they're a larger, more widely ranging snake, and we're basically never getting them in these small patches, whereas we were in the large patches in the core areas. Some species we're still getting across the landscape. So fence lizards, maybe not surprisingly, you're seeing them across the landscape, although even fence lizards were missing in some of these small patches. Side blotch lizards, and in fact some of our highest densities of side blotch lizards were in these smaller patches, so they're still across the landscape. But overall, fewer species in these small patches, more in the larger patches, and then the most in these core areas, which you would expect. So here's just diversity, or actually richness versus area here, so the fewest in the small patches. But I want to mention that that doesn't mean that these small patches of habitat do not have conservation value. So this is if you add up all the species that we found in all those small patches and compared that to the richness that we found in the large patches and in the core areas. And in fact, even though the area here, so the sort of amount of conservation dollar you would have spent there, is half as much as even one of these larger patches, we had as many species in these small patches in them. So it'd be interesting to see over the long run, though. One thing is, what about these small patches? Is it sort of a situation where the species are just hanging on there and over time they're going to be lost? There's a small patch right behind our office where we had whiptails, and we had them early on in the study, and then we came back and started re-sampling that, and just in the last year or so we caught another whiptail and a young whiptail in that patch, and so that's good news that it seems like maybe it wasn't just an ephemeral population there. Okay, so then the other thing, again, we were interested in genetics for these guys as well. So remember so fence lizards and side blotch lizards and skinks are throughout the landscape, but what about, are they potentially being affected genetically? And this is work that Katie Delaney did, who was a grad student here, actually also in Bob's lab, and then was a postdoc for me here, and now runs all of our reptile and amphibian programs for the Park Service. And so we were interested to know, for these species that are still widely distributed, are they starting to be affected genetically? And then actually we were interested, and Katie knows a lot and is really interested in birds as well, and hopefully we'll get some bird stuff going at the park soon too, but she actually went out and misnetted wren tits. So you think that, okay, birds fly around, they shouldn't be affected by erodes and fragmentation, but if one was potentially going to be affected, wren tits was a good likely candidate because they're pretty tied to certain habitats and they're not super willing to fly back and forth across open areas. And so we went out and we had samples from all the pitfall trapping, and then Katie misnetted in all these places as well. And we found, again, so we were going to show these structure slides, we found significant genetic differentiation between these little patches of habitat for all of these species. So here you can see with structure you have these different genetic clusters, so 5 clusters for side blotch lizards, and you can see a number of these clusters are in specific little habitat patches, distinct from anything else genetically. The same thing with skinks. So again, a number of intense differentiation associated with these fragments. Same thing with fence lizards. So again, like this green one here and this blue one here. And even with wren tits, we saw some significant clustering. So this yellow cluster over here, the blue cluster here, and then a different genetic group here right next to another one here. And one of the things that Katie also did was to use this program called Alleles in Space, where it measures the amount of genetic differentiation across the landscape. So the way she did it here is the red colors are more genetic differentiation. And she found very consistently, so this is a freeway here, but this is also the oldest and widest area of development. And you can see the most differentiation here for side blotch lizards is across that area. Same thing for fence lizards, same thing for skinks. There's a little bit of an area over here too. And the same thing for wren tits. And then if you look at all 4 species together, they all have this pattern of the strongest genetic differentiation is across this oldest and widest area of development, where there also is a big road here. So overall, we also, I didn't go into the FST values of the variability, but overall, we saw significant divergence in all 4 of these species, even though they're pretty different. I mean, 3 of them are lizards, but skinks are very different ecologically and taxonomically, and one of them is a bird, right? So very different species with a similar pattern. And actually, we saw reduced genetic variability in them as well, increased relatedness in the lizards, and fewer alleles in the wren tits, just like we were seeing in the mountain lions. One thing that'll be interesting to see, so we've actually, in the last few years, we've redone our sampling program, so now we have 80 sites all over the Santa Monica Mountains and Simi Hills, but we have kept 20 of these original sites, including 9 in the Simi Hills, and so it'll be interesting for these 20 that we're still doing, it'll be interesting to look and see what's going on 10 years ago versus now. there's been some concern about species like fence lizards where they've had declines related to climate change. And actually, we've seen some evidence, just recent comparisons Katie was making between now and 10 years ago, that maybe significantly fewer captures of skinks than we saw even 10 years ago. So that'll be another interesting, sort of thing that's valuable about having this long-term perspective. Okay, the last thing I want to talk about is stream surveys. So we're also interested in aquatic amphibians, not nearly as much diversity, but some potentially interesting things going on with them. So we do these stream surveys, we characterize the habitat, we count all the amphibians, we count the adults, we look for reproduction, so like an egg mass like that for newts, and then we look for the tadpoles. And then we're also concerned about invasive aquatic species like crayfish here. The first 5 years or so of the study, we did basically together with our partners, Pepperdine and the Resource Conservation District, all the major streams throughout the Santa Monica Mountains. And again, we're interested in what are the potential effects of urbanization. And so using GIS for each of the watersheds, so this is a survey point here, above the watershed, how much of the watershed is developed. So this is in Arroyo Sequoia, which is almost entirely undeveloped. But then we had other watersheds like this, like Medea and Ladero Canyon here north of 101 in the Simi Hills, where very high percentages of development in those watersheds. So what effect does that have on the streams and on the biota? And actually we found that with only about 8% urban development in the watershed, we saw impacts on the streams and on the biota. This is just sort of a picture of what that looks like. But here you can see, and again, don't worry about the details here, but here's all of our streams from that inventory period. This is that 8% line here. These are the native amphibians here, so newts and California tree frogs and Pacific tree frogs. And then these are the introduced species, crayfish and exotic fish. And you can see the introduced species are basically only in these urban streams over about 8%. And then a number of the native species like newts and California tree frogs are only in these less urban watersheds. Some species like Pacific tree frogs are throughout the landscape, although even these guys, if you look in a little more detail, can be majorly affected by especially things like crayfish. So this is just what that looks like. So you can see newts much more in the natural streams than the urban ones, and then the crayfish, which come from the southeast, are basically only in the urban streams and only rarely, and even there probably because of other extenuating circumstances, in the natural streams. And then as I said, we see these habitat differences. So we characterize the streams, the different habitats as run, riffle, or pool, and you can see in the natural streams there's a lot of diversity. So there's pool, riffle, run, riffle, and nothing is too long, whereas in these urban streams you get these long stretches of just runs, so relatively low diversity of habitat. And it affects other things too, like stream invertebrates. So more recently, that was the first few years was that inventory period. Now we have 56 different sites that we're monitoring all over the mountains again with our partners. So we randomly selected a bunch of new sites. But reassuringly, we're seeing the same pattern. So now through 2010, so another 5 years, and with all these new sites, we're still seeing this strong effect where the newts and the California tree frogs are basically only in the natural streams, and then the exotics, like the crayfish and the fish, are really just in the urban streams. Just very briefly, another thing that's sort of interesting about being able to do this long-term work is, as you know, we have major fires regularly in Southern California, and that can have a big effect on the landscape, especially on streams. So this picture is actually from Griffith Park, but you can see you've got all kinds of sediment in these streams after these major fires. We had a major fire in the Simi Hills in 2005. So this is one of our streams that we monitor for amphibians, and you can see most of the watershed burned, and it had a big effect on the stream and therefore on the amphibians. So this is average pool depth here from 2000 through 2005, and you can see it just totally dropped off the table. In 2007, there were basically no pools whatsoever. And then this is the number of habitats where we found newts and the percent of habitats where we found newts, and you can see lots of newts in lots of different places up through 2005, and then very little after 2006. This is newts in one of the sort of non-pool areas that was remaining. One of the things that happens, so this is a major newt breeding pool that was 4 or 5 feet deep and had lots of egg masses every year when we sampled it, and it got filled in, and then it got filled in by cattails and then by willows, and this is what it has looked like ever since. Although there was a pool right below it here that was very deep, and that one actually got scoured out again and is still used by newts. So one of these things that you can see over this long-term study. So the last thing about streams is we're pretty excited about some recent work we're doing with red-legged frogs. So this is a red-legged frog. They don't all have as red legs as that. But red-legged frogs are a threatened species, federally listed, that used to be all over the Santa Monica Mountains in all the major watersheds, even out here by UCLA. There's historical records. And since the 70s, they've basically only been in one very small population north of 101 in the Simi Hills, in this East Las Virgenes population. And that population is doing pretty well. With some mark recapture estimates that we did with USGS, and it looks like there's about 100 adults there. But it's really the only population, and there wasn't much movement. One of the things that we were able to see through our long-term monitoring actually is in 2009, we found a red-legged frog in the main stem of Las Virgenes, which is one stream over, and so it looked like they had somehow migrated over there. We only saw a few, but we saw a few frogs in there in 2009, 10, 11, and 12, and we actually saw reproduction over there. So that was a new area with reproduction that we were able to document with our long-term monitoring. But overall, like I said, they're threatened, and they've basically almost entirely been lost in the bottom part of the state, and they really were not seen in the Santa Monica since the 70s. And so just this past year, we started to do translocations. So we went out. These are red-legged frog egg masses. There's a bunch of folks. There's Katie. So we collected half egg masses, and we drove them across the freeway to the Santa Monica Mountains. And as of 2014, there are now red-legged frogs again in the Santa Monica Mountains, at least for a while. We'll see how they do. We put them in pens here. So we moved, not that many, because we don't want to threaten the population. We do have 4 half egg masses, about 1,000 eggs, into 3 different sites in 2 streams. Fed them various things for a while, and we had a little bit of loss, but generally they did well. Released all the tadpoles in mid-July. And then we have young frogs starting in mid-July, so we'll see how it goes. But in the long run, we're going to continue monitoring those sites, add a couple more sites, and then continue to translocate sites, egg masses to our current sites, and we'll see what happens. Just one last mention. So there's another graduate student, Catherine Pease, who worked here in Bob's lab, and she was, again, able to expand and add to our work. So she looked at Pacific tree frogs and crayfish and looked at the potential evolution of morphology relative to being introduced to this new predator. So just the last thing, I didn't mention a bunch of other stuff that's going on, but we have lots of different road projects we've been doing. We did a 9-year study on coyotes, and actually a major thing we haven't ever learned about with coyotes is coyote-human conflicts in urban areas, which people have a lot of opinions about, but there's almost no data on. And then birds is a major hole for us. We really haven't been doing much in the way of birds. We'd love to do long-term monitoring, point counts, and things like that. Katie would love to do it as well. So we'll see if we can make something like that happen. Jeffrey Lee, actually, here in the department, is doing some really interesting work along those lines for his master's. And then again, I mentioned Katie as a postdoc, but we have 2 postdocs doing really interesting work, again, building on all of this long-term data. So Eric Abelson looking at animal behavior and roads, and then John Benson doing a population viability for mountain lions. So that's it. I hope I've convinced you that there's some good value in doing this long-term research in one place and learning about a lot of different things. Thank you. Sure, yeah.
Okay.
Yeah.
About managing bobcats. So there is a study that I don't think it's published yet in Canada, but urban coyotes manage. And it shows that they change their behavior in general, but they also cross-road much more.
The bobcats do?
Coyotes.
Coyotes, okay, yeah.
I'm wondering if it also applies to bobcats here. So whether you looked at mortalities, which are not because of, not managed mortalities, but other sources of mortalities, whether these animals had managed or not. Also, it may explain in some way the genetic differences in genetic composition between before and after the mange. It changes their behavior instead of crossing roads more.
Oh, yeah, so whether mange is changing their behavior. Yeah, no, so that's a really interesting question, and something it's tough to get at, but we have tried a little bit. So one thing we think, when these animals get really sick with mange, they come out more during the day and they're wandering around more in urban areas and things like that. I think it's unlikely that they're moving sort of larger distances when they have mange. At least from what we've seen with the bobcats, they seem to be pretty lethargic and seem to be having trouble. But yeah, it's interesting to know. It'd be interesting to look more in terms of what how that definitely is affecting their behavior. There's no question about that. Although in terms of genetically, they're probably not. I don't know that they're reproducing much when they're pretty sick with mange. So, yeah, Dan.
Are there any notable signs of inbreeding in the lions?
There have not been so far yet anything that we've seen in terms of. So you probably know in Florida, right? They had all kinds of issues like atrial septa and males that had one or both testes that didn't descend, and even some pretty obvious things like kinked tails and a whorl in the back. So we haven't seen those, although the Santa Ana folks, they just had a paper come out in PLOS One, and they also had relatively low genetic diversity, and they did have a couple animals that looked like potentially they had kinked tails. So
Lower survival for inbred.
you mean, have we seen evidence of that? Not really. I mean, the young ones seem to be surviving until they try to disperse, and then not so much. So, but that's probably not related to to inbreeding. Yeah, Dave.
Getting back to the mange, you did all these sort of candidate gene approaches to it, and I was wondering if you actually had done things like, or were thinking about doing things like RNA-seq to explore more broadly what was going on genetically, and perhaps find out more about what's going on.
Yeah, I mean, Bob can answer this question better probably, but yes, the answer is yes. We would love to do that kind of thing, and are thinking about trying to, get money. But we actually Laurel was collecting samples even already that could be potentially used for RNA work. So it's definitely something we'd like to do. Yeah.
In progress. We've collected RNA samples from these bobcats. It's part of the problem, but all the samples we've collected in the past were simple for DNA, not RNA.
Right.
We have now collected samples from about 40 individuals for RNA extraction into bonds.
This is blood or
It's blood.
Yeah, Brian.
So, an awful lot of what you've looked at, whether it's on lizards or lions or whatever, is the effects of these barriers. And, structure keeps pointing out over and over.
Yeah.
So, surely that takes you towards a system migration
Mhm.
and the question of whether that should just become a routine management strategy in the park.
Yeah.
Just always go out and
Move things around. Yeah.
lizards and just move them across the freeway.
Yeah. Yeah. No, it's true. I mean, we haven't thought about it too much yet for the smaller things, for example, but it comes up all the time with the mountain lions. And as many folks probably know, we're working hard to try to get some kind of a crossing over 101. I mean, I guess our hope is that, we don't get to the point where we have to do a lot of that, because it's sort of more manipulation than we'd like to do. But we would certainly be open to it, from everything from, lizards to mountain lions. I mean, say we don't get much in the way of a good crossing over 101, and we continue to see issues, and we start even to see, some actual genetic defects, then yes, I think we would absolutely be willing to move animals around. And in Florida, it was incredibly effective. It basically totally turned that population around. So, yeah,
So,
Ryan.
I guess the other side of that coin is, if you could have sustainable populations on either side, it seems like the 101 is a good sort of genetic, creating good genetic diversity. And if you had manageable populations on either side, and you could conserve both of those, for instance, under climate change,
Like for smaller things,
you're
yeah.
increasing your amount of genetic
I guess that's possible too, yeah.
And I wonder,
How are you increasing it?
well, you're having a sort of a barrier to genetic diversity, so each one of those populations is evolving now separately.
But I guess it's a little-
Between the less within. I mean, you're not creating new wheels, but
Well,
you're just partitioning it.
you have two
Yeah.
lineages now that are,
Yeah, but
I mean, it's a little bit of a thing where you have to be
for
playing.
mountain lions, I don't think it's an issue, but for small things that could have sustainable populations both north and south of the 101.
Yeah. I mean, it's a little bit getting into sort of more manipulation than we want to be. But anyway, it's interesting to think about. Yeah.
Was there any adaptation that we saw that prevented further mange in the bobcat populations? Is it like a density thing? Are they going to collapse again at some point?
You know, that's an excellent question. We were just discussing that actually because, as I mentioned, the population has been coming back in some of these areas, right? And yes, lots of diseases are density dependent. And so we do wonder, I mean, a lot of stuff has been happening, so we definitely think there's this association with mange and rodenticides. And there's a lot of stuff happening to reduce the use of these rodenticides, especially the second generation, I didn't get into this, but the second generation ones, which are the most toxic ones. Although there's still potentially a fair amount of use because there's some restrictions that haven't occurred, like they restrict use by homeowners, by the public, but then licensed providers can still use them. So yeah, so if the bobcats stay in the same area and there's still rodenticides and there's still mange, you know, there could well be another, especially if the density goes up like that. So it'll be interesting to see. One thing that we'd love to have is some information on how prevalent mange is out there in the environment. So a serologic test for mange would be awesome. And actually we worked with some folks up at Davis to try to make it happen, and it hasn't happened yet. So, but that would be great just to know, like, we assume that these animals are generally exposed to mange all the time and that they only really get sick with it when they're compromised by something else like rodenticides, but we just don't know. The only time you find mice is on extremely sick animals, basically. So yeah. Okay, yeah, Paul.
Practical question.
Yeah.
So even if you just walk outside the building right now, you're going to find
Yeah.
rat bait stations
Yeah.
all over.
Yeah.
And you see them, they're ubiquitous
Yeah.
throughout this environment. So, I mean, what should people be doing, you know, instead? Because there is a need to control the rodent populations for public health reasons, but obviously it's having negative impacts on the local wildlife.
Right.
I mean, what would you like to see as a park manager?
Right. I mean, that's a good question. You know, to some extent, I'm not I'm not a rodent control expert, so I don't know a lot about that. I mean, I think a lot of situations actually can be controlled much better by doing preventative type things, and no rodent killing is required. But there are a lot of other ways that you can do it other than poisoning. You know, various mechanical means like snap traps or live traps or rat zappers, which are these little traps that electrocute animals. But you know, I think in the long run, the most important thing is to try to figure out why you have the problem and eliminate that problem. And it's true. I mean, there's probably always going to be some amount in restaurants or various places of rodent control that has to occur. Although most of those places are probably not places where there's going to be as much movement out into the natural areas. And so I think it's really park neighbors that we need to do a lot of education and hopefully have them change what they're doing. So,
Thanks,
all right,
Greg.
thanks.